Our first speaker is Professor Naomi Allen. Naomi is a professor of epidemiology at the Nuffield Department of Population Health at the University of Oxford, and she joined UK Biobank as a senior epidemiologist in 2011 and 2019. From 2019, she's a chief scientist for UK Biobank, and she's responsible for coordinating the future scientific strategy, the linkage of participants' follow-up with electronic health records, and the web-based questionnaires. Her own research focuses on the role of diet and obesity and biomarkers in cancer development. Naomi, thank you for giving us your time today, and we look forward for your talk.
Thank you very much. Let me just share my screen. Okay, so I'd like to talk to you today a little bit about how UK Biobank was designed in order to enable research into a whole range of chronic diseases, including neurodegenerative diseases such as dementia and Parkinson's disease. So first of all, there are various aspects about setting up health resources which make them particularly suitable to study the potential risk factors and causes of various diseases of middle and old age. So the first is for a type of observational study to be prospective. So by that, I mean that it's important that the measures that we want to look at, whether that's lifestyle or physical activity, sleep, biomarkers, are collected many years prior to the disease being diagnosed, in order to avoid bias, with a long duration of follow-up of all the participants to find out what happens to their health over time. And it's in this way that you can really enable a robust research into what the risk factors happen at recruitment and how they are then related to disease many years later. So the second main design characteristic for a study, a prospective study like UK Biobank and many others, is they have to be large. So UK Biobank recruited 500,000 participants about 15 years ago. And the reason why prospective studies have to be large is that only a very relatively small proportion of individuals will develop any particular given disease over the time of follow-up, because we're recruiting people from the general population. So that's the reason why 500,000 participants are needed in order to really do robust research into many common diseases of middle and old age. So in UK Biobank, we've also collected very detailed information on all of the participants. So we've characterized them as much as we possibly can. At baseline, over 2,000 individual measures were collected. Now with continual data collection, it's more like 8,000 or 9,000 discrete different data points that we have on all of our participants, meaning it's a very rich resource in which to study lifestyle and genetic factors in relation to disease. So another aspect of the design consideration is that all the risk factors or measures that we wanted to collect on individuals were done on as many participants as possible. So we were comprehensive, but also that the data that we were collecting is of high quality as possible to enable the most high quality research to be done. So while the UK Biobank participants are not representative of the UK population as a whole, we made great efforts to ensure that the participants have a wide variability of a range of different exposures, whether that's relating to socioeconomic status, physical activity, dietary habits, and so on, in order to enable research into how variation across these different risk factors influences subsequent disease risk. In addition to characterizing our participants as much as possible in relation to risk factors, we've also tried to do the same for their health outcomes. So having comprehensive and detailed information about people's health over the follow-up of the study. We also have high participant engagement. We ask all of our participants to do follow-on questionnaires, come back for further in-clinic assessments such as imaging, with over half of the study cohort willing to do so. And perhaps most importantly, and I'll come back to this at the end, is that all of the data that has been collected is easily available for researchers worldwide to perform research and to enable other researchers to either confirm or refute their findings. So way back in 2006 to 2010, we recruited 500,000 individuals aged between 14 and 69 at that time across England, Wales and Scotland, around about 22 different assessment centres. So we invited everybody who lived within about a 25-mile radius of each of these assessment centres to come along to join the study in order, and the choice of location of these centres was strategic in order to really increase the heterogeneity. So we included places like Hounslow and Birmingham, Glasgow, as well as London and Oxford and Manchester and so on. As mentioned, we've got extensive information on all of our participants: lifestyle, environmental exposures, medical history, personal lifestyle factors, cognition, hearing tests, eye tests, a whole range of physical measures: blood pressure, lung function, body size, and a range of biological samples: blood, urine, and on a subset of participants, also saliva. And we've got consent to follow up everyone's health through linkage to their electronic health records, and also to go back to them directly to find out what happens to them over time. So we've got a huge amount of data available for researchers to analyse: lifestyle questionnaires on all 500,000, genomic data on all 500,000, including genotyping and whole exome and now whole genome sequencing on 500,000, which is certainly the largest study in the world to have such granular genetic data on such a large scale. A whole range of physical measures, including in a subset of 100,000 physical activity monitor based on a Fitbit type device to obtain objective measures of activity and sleep. As I've said, we follow up all of our participants, their health, through linkage to electronic health records. On a subset of participants up to 100,000, we're doing multimodal imaging scans. And we've also just started a repeat imaging scan on up to 60,000 of those images. It's the largest study in the world by some margin to have imaging data included into a resource like this. And we have a whole range of biomarkers measured in all 500,000, which I want to spend a little bit of time talking you through, because this is where the design characteristics of UK Biobank have really made this part of the study unique. So we've really focused over the last few years our efforts on turning the depletable samples that are limited into reusable data that can be used by everybody. So as I've said, we've got very detailed genomic data collected for all 500,000 participants. We have a wide range of biochemistry markers, measuring things that researchers are really interested in for a range of diseases such as cholesterol, sex hormones, markers of kidney, liver function, full blood count, and so on. We have telomere length available on all the cohort, which is a marker of biological aging. We're currently undertaking metabolomic assays, so measuring circulating levels of metabolites, which is mainly circulating lipids in the full cohort. And we've also undertaken a project to measure circulating proteins in an initial tranche of about 57,000 participants, but hopefully to expand that to a larger subset of the cohort in due course, which covers about 1,500 proteins and soon to be expanded to 3,000 proteins, which will be released later on this year. And this is important because we're probably one of the very few studies in the world where we have cohort-wide measures of biomarkers. Most of the studies have tended to measure biomarkers in a group of individuals with a disease of interest, often called cases, and a group without the disease, often called controls. So these nested case control assays may often deplete the sample more rapidly, therefore you can't do further assay samples on them, and they may generate assay data that's not comparable with the rest of the cohort when those measures are done. So by contrast, performing cohort-wide assays, while it's very expensive to do it up front, actually on the whole is very cost effective because you can do it at scale. It minimizes sample depletion, massively improves quality control of the data. You are comparing data directly from one individual with another in the cohort. They're directly comparable. The data are readily shareable and quickly shareable with researchers, and it can enable a very wide range of research. So while the case control approach in previous studies is often lower cost in the short term, having all of these biomarkers done on the full cohort is far less costly in the long term, and it's something that UK Biobank has been a strong advocate for since its inception. As I've said, we follow up participants over time to find out about their health outcomes through linkage to NHS health administrative datasets. We now have 15 years of complete follow-up through linkage to national death and cancer registries, hospital inpatient admissions, including critical care, COVID-19 test data, and also primary care for just under half of the cohort at the moment. And there are ongoing efforts to obtain these data for the full cohort in due course. And what you can see is that by using these health data sets, there are already thousands of new cases of the most common health outcomes that have been diagnosed, as you can see from the table on the right. And over the next 5-10 years, as the cohort ages, those number of cases of health outcomes increases substantially. So you can see here for Parkinson's and Alzheimer's, by 2032, there's many thousands of incident cases, really enabling robust research into these areas. So I just wanted to touch on the importance of how easy it is to access these data. Because it's so straightforward, it's meant that there are now over 30,000 researchers, both from academia and from commercial companies, accessing these data from around the world. Over 90 countries, researchers are accessing these data, with over 7,000 publications using the UK Biobank data, and some of that research we'll hear about in today's webinar. Over 2,000 of the publications were just published last year alone, so you can see there's an exponential increase in the research output from the resource. By having it so easily available for researchers to use, really maximizes the utility of the resource by the worldwide research community. It enables collaborative research and multidisciplinary research. It enables researchers to either confirm or refute or build on other people's work using the data. And we've really tried to make an effort to make sure that the access procedures for all researchers are simple, straightforward, equitable, and transparent. And I think we're one of the few cohort studies in the world where that is the case. So we had in 2021, we released our research analysis platform, which is a cloud-based analytical platform where researchers, instead of downloading the data onto their own computers, can instead access, store, and analyze the data in a secure platform on the cloud. And this is really starting to democratize access by bringing the research to the data rather than the other way around, and making it much easier for researchers for low middle income countries, for example, and early career researchers to access the data who might not have that infrastructure in their own institutions. In addition, at AWS have also provided free research credits for early career researchers and researchers from low middle income countries. So effectively for these researchers, accessing the data on the cloud is free. And that's really helped to enable use of this type of big data for research purposes by these communities. And having the data on a secure cloud platform also facilitates data challenges, whereby researchers can basically put in an application to do a kind of competition or hackathon or a data challenge in order to get the community to work together on a specific problem. So we encourage you, all of you who are thinking of doing some kind of data challenge, to think about using UK Biobank. So I just want to summarize by concluding on the unique combination of 4 areas that makes UK Biobank particularly stand out. Firstly, the scale, 500,000 participants who were heterogeneity and lifestyle factors who joined, exquisite detail about lifestyle, environment, biological assays, imaging, and so on. Long duration of follow-up over 15 years, yielding very large numbers of incident outcomes to enable robust research, and the ease of accessibility of the data, as I've said. These 4 components or study design metrics of the study have really made UK Biobank a unique resource to do public health research and data discovery. So I'll end it there, and I'll stop sharing my screen. I'm very happy to take questions at the end.
So let's move on with the Q&A, and I'll pick up just a couple of questions and then pass over to Joanna, who can follow up with those that follow. The ones that I'll start with are questions that are largely directed, I think, towards Naomi, and I'll pick out 3 of them to lead off. And Naomi, there's 2 of them are very much related, and one was to ask how representative the 500,000 participants in UK Biobank are of the UK population. And then a not unrelated question is how ethnically and genetically heterogeneous the UK Biobank population is. Maybe you could take that, and then after that, just follow up with a bit of detail on the cognitive measures included within UK Biobank.
Yeah, sure. So the first point around how representative is the cohort. So the 500,000 individuals who joined UK Biobank are not representative of the general population of the same age. They are slightly wealthier and slightly healthier than the general population of the same age. And there was a paper published about this in 2017 by Anna Fry, if you want to look that up. Having said that, for most analyses, and it's worth checking all analyses you do for various types of selection bias, but as long as you have sufficient heterogeneity in the exposure of interest, then any internal associations you find, say between socioeconomic status and Alzheimer's disease, are likely nonetheless to be valid and generalizable to the population at large, even though the UK Biobank population is not representative of the general population. There are various analytical approaches researchers can take to firstly identify and secondly account for or adjust for types of selection bias and collider bias, which deals with this whole representative issue. So that's kind of the first thing. Related to that, in terms of the ethnic diversity of the population, so we try to recruit across a broad range of different areas in the country to get as many Black and ethnic minority groups as we can. The cohort is about 95% white, which was actually at the time, 2006, was pretty much the same as the UK population as a whole. Of course, it's not representative of the UK population now. Having said that, we have whole genome sequencing data on 10,000 people from South Asian backgrounds, 10,000 people from African backgrounds, which is the largest study in the world with whole genome sequencing data. So whilst we have relatively small numbers of people from Black and ethnic minorities, for some types of data, it still represents a hugely valuable resource. And then on the third issue around cognition, so it's worth looking at the UK Biobank Showcase, which is on our website, which shows all of the variables that we have collected on the individuals. We have a whole range of different types of cognitive function tests, both performed when people come along to an assessment centre, but also done remotely as part of online questionnaires. And we have up to 15 or so different tests, ranging from verbal and non-verbal reasoning, prospective memory, numeric working memory, executive function, processing speed, and so on and so forth. All of that is documented on the website, so I encourage you to have a look at that. And it's just worth saying, actually, that we're going to introduce another cognitive function questionnaire developed actually by colleagues at Imperial that it's purported to be a bit more sensitive to look at early biomarkers for cognitive decline. So we'll be rolling that out in due course. Thanks.
All right, thank you very much, Naomi. There are a few questions around sort of the COVID data, which I think are probably for you, Naomi, and Eugene. Naomi, since you started, I can see some questions on the COVID antigen test, that whether those are available, and the COVID test data, and maybe you can sort of quickly summarize the COVID sub-study.
Yeah, so we, during the pandemic, we linked to the COVID-19 PCR test data, which is all available, again, through the data showcase, and we're still linking to that data, England, Wales and Scotland, and we make that data available on a periodic basis. That data should all be there, it's all up to date, it's just a question of finding it on the data showcase. In addition to the PCR test data, we also did a sub-study where we sent out lateral flow antibody kits to as many participants as possible, and over 200,000 participants, we've got data on whether they have antibody positivity to the virus, and whether they've been vaccinated or not, and so on. So again, all of that data is also included in the resource. We're currently just processing data on COVID vaccination. So we hope to make that data available probably in the next 6 months or so, is what we're aiming for, which again should help to, for those who are still interested in COVID-19 research, we're doing our best to try and get that data out there. So yeah, we have a lot of data available on positivity. It's just a question of finding it on the website, but it is all there.
We're reaching the end of the time, but I did want to get back. There were a number of questions about the proteomics data. And very briefly, Naomi, could you comment on the current availability of proteomics data, particularly the Olink data, and when the next tranche of data will become available? And at the same time, you might comment on how the data in UK Biobank, how frequently it is updated, both clinically and with other measures.
Yeah, sure. So the Olink data, which done a pilot study funded by an industry consortium that has measured proteomics using the Olink platform in about 57,000 participants. Data on 1,500 proteins was made available a few months ago. That's now been expanded to the 3,000 protein platform. Those data will be made available, I want to say July. It's either July or October, later this year. Covers both of them. I think we're hoping for October. Yeah, so those data will be available then. We're also thinking about the consortium who have funded this Olink work are also currently comparing different proteomic platforms, Olink, SomaSeer, and then 3 mass spec platforms, in order to inform the best way forward for UK Biobank in when we scale this up, which platform or complementary platform approaches we should be adopting. So that information will become available in due course. And sorry, Paul, what was the second point?
The second question was just a more general question about the frequency of updates to the platform.
Yeah, so it's we try to go for 2 to 3 a year. It's a lot of work to do an update of the database. We try to update the health outcomes at least once a year. So that's death, cancer, hospital admissions, and we're up to date with all of that now. So it is worth saying, though, that we're currently changing our underlying IT processing pipelines for all of the NHS records. So we're having a bit of a hiatus, and once we've got those new tools in place, which may take a year or so, then we'll have more electronic health record data. But certainly in terms of sample assay data, more imaging data, that will come through roughly every 6 months. So that's the plan.